Evening, everyone. Welcome to the UConn panel series on COVID-19, Hope, Health, and Heroes. This is a series of 3 panel discussions. And the first one is on vaccines and treatment. I'm very, very excited to present an outstanding group of panelists, experts on these 2 topics. So briefly, my name is Bruce Lian. I'm the Dean of the UConn School of Medicine. I'm a physician scientist, a cardiologist, and I run a research lab. And some of our research effort has been diverted and focused on COVID-19 as well. So it's an area of a recent passion for us here. And just my pleasure and privilege to be part of this very distinguished group of panelists. And without further ado, we're gonna go ahead and ask each panelist to self-introduce briefly. We're gonna go to our first panelist, Dr. Adrian Hernandez Diaz. Adrian, over to you for a brief self-introduction.
Sure, thank you, Bruce. So I am Adrian Hernandez Diaz, and I am an associate professor of comparative effectiveness and outcomes research at the School of Pharmacy at UConn.
Thank you. We're gonna go next to Dr. Anthony Vella.
Thank you, Bruce. Hi, everyone. Thank you for attending. My name is Tony Vella. I'm the chairman of the Department of Immunology. I'm also the Senior Associate Dean for Research in the School of Medicine. And my laboratory has been studying all things T cells for about 25 years or so. And it's very exciting to be on this panel and tell you more about some of the great and interesting work that's going on in this, in the role of T cells and COVID immunity. Thank you.
Thank you, Tony. We're gonna go to Dr. Jen Geraldo.
Thank you. I am Jen Geraldo. I am a clinical associate professor at the School of Pharmacy. I also am passionate about immunizations. It's one of the areas that I really have focused over the past 15 years. And thus far, we have trained almost 2,000 pharmacists as immunizers over this time period as students or pharmacist immunizers.
Thank you so much, Jen, for everything that you do. Last but not least, Dr. Kevin Deekhouse.
Good evening, everyone. So my name is Kevin Deekhouse. I'm an associate professor of medicine and head of the Division of Infectious Diseases. And first and foremost, I'm a clinician. Prior to COVID, had an interest, still do, have an interest in HIV, travel, and tropical medicine, and have been involved in our epidemic and pandemic efforts, including swine flu and response to Ebola. But with COVID, it's been a lot of direct patient management and institutional response to COVID, including development of our biocontained units, treatment protocols, and really clinical research protocols. It's good to be with you this evening.
Thank you, Kevin. So the format today is gonna be a series of dialogues. I'm gonna be the moderator, and I will pose questions and initiate a dialogue with each of our 4 panelists. And so first of all, so everybody knows this, but COVID-19 is now the leading cause of death, surpassing heart disease, which was the leading cause of death in the US. And we know that the risk differs depending on the person. Some of them are very prone to having a bad outcome. And we now know that the risk of severe disease leading to hospitalization and about anywhere from 3 to 5% of the people will actually die once they are in the hospital. And the risk of age, there is almost a linear relationship between the age and the severity of the disease. Underlying medical conditions such as diabetes, heart disease, heart failure, hypertension, lung disease, cancer, and actually obesity. So there's also almost a linear relationship between obesity, the degree of obesity, and the bad outcome from COVID. We as a community have learned a lot, but much more is needed. Right now, COVID has afflicted 25 million people in this country, but we actually think close to 50 million Americans have been infected with this contagious disease. And that's because almost half of the people walking around have minimal or no symptoms. So they don't even know they had the virus, but nevertheless, they did. And that's why 25 million are those people we know because of their symptoms and tests, but probably 50 million of them actually have the disease. We're going to talk about the vaccines. We're going to talk about the treatments, and we're going to learn firsthand from healthcare workers at the front line delivering care and treatment to patients with COVID. So our first panelist is Dr. Adrian Hernandez Diaz. Adrian, thank you so much for being here. Can you tell us about the kind of medical treatment such as remdesivir, convalescent plasma? There's a drug called the interleukin-6 inhibitor for COVID-19.
Yes. Thank you, Bruce. So one thing I forgot to mention was that I've been involved in the last few months in evaluating systematically several interventions and treatments directed to treat patients with COVID-19 and to avoid people getting infected too. And among those treatments are the ones that you are mentioning. So remdesivir, convalescent plasma, and tocilizumab, one of the anti-interleukin-6 drug, are three of the treatments, interventions that we evaluated systematically. By systematically, I mean formally in systematic reviews. And remdesivir is the only drug that was fully approved by the FDA for the treatment of hospitalized patients with COVID. However, the publications and the studies available about the treatment with remdesivir are not solid enough to recommend clearly in favor of using remdesivir. Even though the FDA has recommended remdesivir, the main issue is that the follow-up of the studies is pretty small. There are 2 randomized controlled trials evaluating remdesivir against placebo, and there are 2 more trials comparing different doses, different times of treatment with remdesivir, and one of them compares 2 doses, 5 days to 10 versus 10 days, and versus the standard of care. And when evaluating those 4 studies overall, then the effect of remdesivir on all-cause mortality or need for mechanical ventilation is not seen. So what has been found is a decrease in the time to recovery in patients with COVID-19 and hospitalized. The overall quality of evidence for remdesivir in all outcomes, clinical or intermediate outcomes, has been at the most low. So it's either low or very low. So I'm afraid that the evidence so far for remdesivir has been going in the direction of not recommending it. In terms of convalescent plasma, has been something similar. our overall conclusion of our systematic review has been negative also. We evaluated both randomized controlled trials and cohort studies. And among the randomized trials, we haven't seen a clear effect on clinical outcomes with convalescent plasma. I recall reviewers of a journal where we submitted our study and has been accepted already. Reviewers and the editor were concerned about the, and I quote, the clear efficacy of convalescent plasma in COVID-19 patients according to their assessments. And their comments were based on a very large case series that was conducted at the Mayo Clinic in Minnesota. And the issue with that study is that even though it was conducted in thousands of patients, was a case series. That means that there were only patients with COVID-19 and all of them received convalescent plasma. And the main conclusions for the emergency approval of convalescent plasma in the US were based on subgroup analysis of those patients in that case series. I think were 32,000 patients with COVID-19. But formally, the randomized trials haven't shown positive effects on clinical outcomes with convalescent plasma. And one of the questions that we discussed, Bruce, with you in the last few days was about the timing of the administration of convalescent plasma or the amount of antibodies already in the plasma of patients. And with the few randomized controlled trials that we evaluated, what we saw was that there were differences in the timing of administration, were differences also in proportion of patients with antibodies already at baseline, but we couldn't identify any clear relationship between higher or lower efficacy according to the timing of administration or the presence of antibodies in patients. And finally, about tocilizumab, the anti-interleukin-6 drug, we are in the process of analyzing data. We have finished the main analysis so far, and we had information on 7 randomized controlled trials, including one that was published last week at the BMJ. And we had 9 cohorts of the best quality available. That means that those cohorts properly adjusted for differences between the patients of characteristics of patients receiving tocilizumab and patients not receiving tocilizumab. And we have evaluated several clinical outcomes and also adverse events associated with tocilizumab. And again, we haven't found a benefit on clinical outcomes in randomized trials or in the best cohorts available. So that is the only signal that we identified was that patients getting tocilizumab had higher risk of neutropenia in comparison to patients getting a standard of care or placebo plus standard of care.
Thank you, Adrian. Actually, we have a question coming up on the audience. What is convalescent plasma? How do we get those? I know that you talked about the evidence for efficacy of convalescent plasma. Nevertheless, it's used quite oftenly. So what is convalescent plasma? How do we get such plasma?
So that is the plasma that is processed in the lab and is taken from patients with COVID-19. So what the studies are reporting is that what type of patients of individuals with COVID-19 where this plasma was taken from. So across studies, the amount of antibodies, what is a way to quantify the amount of antibodies that is called a titer. So the titers have been different also across studies. So so that's another reason of these differences among the studies. So formally, plasma taken from donors already who already had COVID-19 with antibodies in their blood, and the plasma from that blood is taken and administered to patients, to other patients.
Thank you, Adrian. So a very famous drug was touted by a former president as being efficacious. That's hydroxychloroquine. However, many studies, both observational and randomized trial, at least in the hospitalized patients, shown that hydroxychloroquine is not effective, and in fact, it's not recommended for use. What about in the outpatient setting? Is there any data to show hydroxychloroquine could be of any benefit in prophylaxis, after being exposed, or mild disease that doesn't require hospitalization? So what, is there any update on hydroxychloroquine you can share with us, Adrian?
Yes, so those are two studies that we completed recently, and we submitted for publication a month ago or so. And our two studies, one is in patients with COVID-19 and in early stages of disease. So that means that those patients were not hospitalized, as you mentioned, Bruce. And we have another study evaluating hydroxychloroquine as prophylaxis. And there are, I think that overall are bad news. One source of bad news is that there were just a few studies, just a very, very few randomized trials in both studies. And there were also a few cohort studies. And we evaluated hospitalization among in the first study of early treatment, and there was no difference between hydroxychloroquine and the control group, and there were no difference in other outcomes either. So about mechanical ventilation or need of ICU, and there was no difference also between adverse events. And in the prophylactic study, we saw that there were two main sources of studies. There was one that is called the pre-exposure intervention with hydroxychloroquine, and those are studies with longer follow-up and higher doses in comparison to the post-exposure hydroxychloroquine strategy. And overall, we did not see any effect of hydroxychloroquine on COVID-19 infection, and we didn't see any difference between the pre-exposure and the post-exposure strategies either.
Thank you, Adrian. So this is a perfect segue to the next panelist, who will talk about some of the research that's ongoing to advance the treatment for COVID-19. Before going to Dr. Vela for that panelist, so thank you very much, Adrian. An audience said that it appears that the three drugs you spoke about aren't showing any true benefit. Is there any emerging treatments that are anticipated to be effective? I think we're going to answer that perhaps towards the end because we have other panelists who might weigh in on some new treatment modalities. But I think the bottom line is that much more work is needed, certainly on the treatment front. So with that, I'm going to turn over to Dr. Vella. So Tony, thank you again for being here. I got a couple of questions for you. So there has been active research to develop new treatments and vaccines for COVID-19. As an expert on immunology, what are some of the examples of research advances you could tell us about? So these are maybe still in the pipeline, in the research lab, and maybe moving to the clinical phase. And any example you can share would be very much appreciated, Tony.
Sure. Thank you, Bruce. I think what I'd like to do is give an example, but also some explanations on some of the terms that people are reading about, hearing about in the news, because it's very important to understand what these words actually mean and how they're being applied to COVID treatment. And to be honest with you, with a lot of other diseases as well. I think one that keeps popping up is monoclonal antibodies. And you'll be hearing, you probably already heard about it around Christmas time, and you're going to hear more and more about it. And what monoclonal antibodies are, they are proteins that people make, that we all make, and we use a lot of mouse models to actually study monoclonal antibodies. So they're proteins that are released from a cell in the immune system called B cells. And when they're released, these proteins will actually bind other proteins. So for example, these specific monoclonal antibodies will bind the spike protein on the SARS-CoV-2, which is the virus that causes COVID-19. And many years ago, the Nobel Prize was awarded for the production of monoclonal antibodies. It was probably one of the greatest breakthroughs in the 20th century in all of science, because it allowed us to have tremendous specificity of how these antibodies could react with different antigens. And I just want to give you one example. You could imagine when you have a ring with hundreds of keys on that ring, but yet each key is unique and will open up one lock. And an antibody is a monoclonal antibody is very similar to that. Sometimes the key might fit in a little bit, not fit in at all. Maybe it'll fit in and not turn the lock, but there'll be one key that'll fit into the lock and then lock the door. And so monoclonal antibodies are very much like that. They are just a variety of different specificities, sometimes very different from each other, and sometimes very subtle differences. And so we can generate these monoclonal antibodies in laboratories, and a number of major large pharma companies have set out to do this probably from the very beginning of the pandemic. And several have received early authorization use. There are a number in line right now that will be used to treat patients. I think some of them actually are being used to treat patients, and there'll be more antibodies, monoclonal antibodies produced that can actually bind to the COVID-19 spike protein. And the power of this is that the monoclonal antibody is very precise. So it'll bind that spike protein on the virus and neutralize it. In other words, that virus will no longer be able to infect the cells of the body. At the same time, this monoclonal antibody will not bind your own cells. That would be something you really wouldn't want. I mean, you certainly wouldn't want to take a drug and then have it bind it to normal tissue in the body. You want it to really seek out and bind the virus. And that's exactly what these monoclonal antibodies do. The important part is that they're neutralizing. That is, that they'll prevent the virus from infecting the host cell. And that's something that is crucial in terms of treating COVID-19. So the other part to this, Bruce, that's very important is that these antibodies have to be given during the viral infection. In other words, as a patient is recovering, let's say 2 weeks out when there's no virus potentially inside the patient, they would be useless. So as somebody gets infected, as they have a fever, these monoclonal antibodies are going to be really instrumental, especially with elderly patients and on down the line. They could be something that would keep people out of the ICU and prevent hospitalization. So that's a really exciting front in the field of COVID research. But it's important to know that these monoclonal antibodies have been used in medicine. They're used in treatments to actually neutralize proteins of our body that we want to keep suppressed. And actually, one of them was already mentioned, IL-6. So we have neutralizing antibodies against cytokines that are also really important to prevent an overactive immune response. So it's not something that's out of the blue. It's something that's been tried and true and is now being utilized to treat COVID patients.
Thank you, Tony. So you're very modest. You're one of the world's leading experts on T cells. These are immune cells in the body. What did they do? Why did they appear to be important in the fight against COVID-19? And what kind of research might be conducting to study these T cells?
Great, that's a great issue to bring up, Bruce. When the pandemic started out, I was feeling kind of like left out of the story because it was all about B cells and antibodies, but all of us T cell immunologists, we knew that T cells will turn the corner and become the star of the show. So you're really not going to get a very good immune response without T cells. I mean, the T cells, they're the quarterback of the immune system. So we're getting close to the Super Bowl. Unfortunately, my team just got knocked out yesterday, so I'm kind of feeling bad about it, but we'll be back next year. And the quarterback is really the key. You're not going to have a very good team without a great quarterback. So T cells, especially T helper cells, they help B cells make antibodies. They help other cells of the immune system attack and destroy pathogens. And T cells turn out to be very essential in the COVID response. We knew early on from actually one of the very first papers that was demonstrated from a patient in Australia. The investigators found a very specialized kind of T helper cell called a T follicular helper cell. And this is a subset of T cells that specializes in helping B cells produce antibodies, these antibodies that we talked about earlier. So T cells help B cells make antibodies, and they're essential to fighting the infection. So when you have Dr. Lanning had mentioned a wide spectrum of symptoms. You'd have patients that are asymptomatic to the point where patients are in the ICU and in very serious state. The antibodies that can be produced, they won't be produced very well without the activation of the T cells. And in fact, what we're finding, which is very interesting and been published in a number of reports right now, is that T cells that were specific to other coronaviruses, some of them the common cold, you may have heard of this, were actually cross-reacting with the SARS-CoV-2, the novel coronavirus that we're fighting right now. So the T cell is really essential to the battle. And I just want to mention something else about the vaccine. I can talk a little bit about the intricacies of the vaccine, but one important point is that immunological boost, the second shot of the vaccine, is crucial to boosting T cell responses. So in some people's eyes, you might think, well, you get one shot and then a second shot, it's kind of like 1 + 1 = 2. But in terms of the T cell response, you couldn't be further from the truth, actually. That booster shot makes it 1 + 1 = 12. You can think of it that way. It's so important to have that booster shot. And that's really what distinguishes immunology from all of the other systems in the body, that booster response where the T cells really take off, provide help to many different arms of the immune system, and provide the protection that we enjoy with the power of vaccination. I'll stop right there, Bruce.
Well, thank you, Tony. So maybe if I can recapitulate what you just talked about in the immune system and its important role, is you got the B cells, which produce the antibodies, which can bind to the spike protein of SARS-CoV-2, which is the virus that causes COVID-19. And that this B cell producing the antibody is like the business arm, because you can neutralize the virus, you can eliminate the virus from the body, you can prevent harm before the virus causes all the damage in the body. And then you have the T cells, which helps the B cells make the antibodies. So if you don't have the T cells or your T cells are not working well, you're not going to produce a lot of so-called neutralizing antibodies. Is that kind of in a lay language term, how the immune system try to hang together to fight this infection?
Yeah, that's a nice summary of how the immune system works. The aspect of T cells being so important is really borne out in the elderly, because many times they may not have very strong T cell function or appropriate T cell function, and that can lead to some dysfunctional immune responses. The other aspect of the T cell response, you've heard about the T helper response. Those are CD4 T cells. The other T cells are the CD8 T cells. And these cells will actually seek and destroy virus-infected cells of the body. So if you have a SARS-CoV-2, the virus that causes COVID-19, infect an epithelial cell, these CD8 T cells will seek those cells out and destroy them. And they've turned out to be a very important population in fighting the infection, something that's really exciting. And actually, another aspect of the vaccine is actually studying how well those cells are boosted in response to the vaccine, because a virus infected cell will replicate and then infect other cells. So the sooner you can get to it to destroy that cell, the better off the patient will be. And that's what CD8 T cells provide as well. So without T cells, you're not going to make a very good antibody response, and you're going to have a poor ability to kill virus infected cells. So they really are the main players of the immune system. And of course, the B cells play a really strong, what we would call an effector role in releasing neutralizing antibodies.
Yeah, thank you, Tony. You just brought up two very good points. I mean, I think we know age is a risk factor for bad outcome. And if age impairs the production of appropriate amount of T cells, that could be an explanation why the older people can't fight off this infection as easily as a younger person. On the other hand, the kids who, when they go to school, they get exposed to all kinds of viruses that cause cold. And as you mentioned, the common coronavirus, which are relatively benign, cause about 40–50% of the cold. And so the kids, they may have already developed the T cell because of the exposure to these common coronavirus. And so they are somehow already immunized in a way against the more serious novel coronavirus, COVID-19. So we just got one additional question before we move on. How much is known about the duration of immunity after natural infection? And I'm not going to ask you this question because I think we actually don't know, but your prediction as to the duration of immunity after vaccine, which is the $64,000 question.
Right. That is the $64,000 question, and it's something that we don't know the answer because we just started vaccinating people. But we know from other systems that that vaccinations do last for a relatively long period of time. It's not, you wouldn't measure it in weeks, but you could certainly measure it in months, maybe even years. But I want to point out something that's really critical in this whole story. If you step on a nail, you run to the doctor and get a tetanus booster. So we may be at the point where we start thinking about booster shots to kind of enhance the vaccine that you got in the first place. Maybe next year, that might be a good idea. As we know how strong the immune response is after the vaccine, my prediction is, I'm just thinking out loud, I think it's going to last for a long time, but they're probably going to need a booster response at some point. And that would be the idea, to keep boosting the immune response and keeping the antibody levels high, but also making sure that we have what we call memory T cells. And that's what vaccines are all about, memory T cells. They have a memory of the response, and then they can come back. In mice, it can actually be years, and in people, it can be decades even. I mean, there's many different studies out there that are obviously non-COVID studies, which look at memory T cell responses and how important they are. And that's actually the basis of the vaccine. And that's what the booster shot would do, would come back and stimulate the memory T cells that are generated from the vaccine that we get now, perhaps next year, a year later, et cetera. So it's going to be really exciting to watch. And I think we're in good hands with the vaccine at this point.
Thank you, Tony. A very exciting potential development and also a perfect segue to our next panelist, Dr. Jen Gerardo. So Jen, I have a few questions for you as well on the vaccine. Well, can you tell us about the two vaccines that currently have been authorized by the FDA under the EUA, which stands for Emergency Use Authorization, and what the expected efficacy is for each, and in the general population, for example?
Sure, thank you so much. So both of those vaccines that have been authorized, not approved yet, but authorized by the FDA for emergency use authorization are the ones that you've probably heard all about. It's your Moderna and your Pfizer BioNTech. Those two vaccines have been shown in clinical studies, and this is really important, and it goes in to state what Dr. Vela said earlier, is all of the efficacy that has been studied for these is 2 weeks after the second dose. And so 2 weeks after the second dose of both of them, they are both very similar, and they have about 95% efficacy. So that essentially, of your patient population, 95% of them will be protected against moderate to severe COVID-19 disease. And that's really what these two vaccines are looking at. They have not studied, and we do not know if they are going to prevent asymptomatic infection. So it is very important that it prevents that hospitalization, severe disease, and death in the vast majority of the population, but you still need to wash your hands. You still need to keep social distancing, and you will still need to wear a mask until it's determined if they prevent that asymptomatic infection.
Thank you, very, very helpful. As someone who has been actively planning and actually coordinating and giving these vaccines, can you tell us about the actual process of storage, the process of making it ready for injection during the rollout? As you know, since the first week or the second week of December, the Pfizer and then the Moderna vaccine have been rolled out. So tell us about on the practical sense, as our patients go to a healthcare providers or eventually a pharmacist or pharmacy, what is the process? Can you just tell us more about that?
Absolutely. So the storage and handling of these are very particular. The Pfizer is the one that needs the most detailed focus. It does need to be stored at a very, very cold temperature. It cannot go in a regular freezer. So it either needs to go in an ultra cold storage freezer, which some of the hospitals have, or it needs to be shipped in a special thermal regulator, of which the institutions have to get a hold of pellets of dry ice. It needs to be changed within 24 hours of it arriving, and then needs to be changed every 5 days. As if that is not enough, it can only be open twice a day for a maximum of 3 minutes each. And it does require special tape to be holding that down because it needs to be at that specific temperature. Once it is defrosted, either in the refrigerator or at room temperature, it then can only stay in the refrigerator for a maximum of 5 days. Once it is taken out and actually gone in to get it for our patients, you only have 6 hours to use that. So it's very important to have all of your patients lined up so that you can use every single dose that you're able to get. Additionally, when they first came out, they said there were 5 doses. Some people, and most people, have been able to get 6 doses. Every now and again, someone will get that unicorn and get a 7th dose out of that as well. But if you do get that luck to get that 7th dose out, you want to be able to get another patient to vaccinate, because every single patient that we can vaccinate is going to help save another life. For Moderna, it's a little bit more stable. And so they actually did update after the initial emergency use authorization, they actually did make it a little bit easier to use. So now it does still need to be frozen, not at that deep ultra cold temperature within the normal freezer range, but a little bit more narrow of a value that you need to keep on top of your freezer for. But once it's defrosted, that can stay in the fridge for up to 30 days, which is a huge deal. The fact that you can keep it in the fridge for up to 30 days, and then once you take it out of the refrigerator, you have 12 hours to use it. Similar to the Pfizer, once you go in that, you only have 6 hours. And so that one's a little more complicated because now you have 10 to 11 doses that you can get out of that. So again, you need to have your patients all set up. And if people don't show, I'm having some sort of a backup so we can utilize all of those. And then I know, Dr. Liang, you're also interested in knowing a little bit about the possibility of that Johnson & Johnson one coming down the road. And I'll go a little bit into that ahead of time, at least with regards to the storage, is one of the reasons that people are so excited about that potentially coming down. And we can talk about what we might or might not know about that in a minute. But the important part of that one is the fact that it's traditionally stored just like every other inactivated vaccine that we have on the market. It's refrigerator. It can get shipped just like the refrigerator other vaccines. It would be more traditionally in line with everything else we usually had done. So it could be in the doctor's office fridge as well as in the community pharmacy fridge. It can be given at so many more sites.
Is there any preliminary data on its efficacy? I guess we don't know the clinical efficacy defined as percentage of patients got the vaccine will not have severe disease. But what about the antibody level that's produced by this Johnson & Johnson vaccine?
Absolutely. So it does look, the preliminary data was published just earlier this month in the New England Journal, and they did show that it did have adequate antibody responses as well as those T cell responses that Dr. Vela had spoken about earlier after one dose and after 2 doses. They then went on to look at the clinical outcomes. And we know that they have enrolled 45,000 patients looking at 18 to 55, and I'm sorry, 18 to 60, they changed the age for the clinical study, and greater than 60 in looking at those durations and clinical outcomes. And they're looking at moderate to severe COVID as well. And they're looking at primarily at 14 days and 28 days. That has closed to enrollment and they're analyzing that. And we are all waiting at this point. We're hoping those will have good outcomes. And we're waiting for that to potentially get submitted to the FDA, hopefully any day. That being said, is that with prior adenovirus vector vaccines, they have shown that a second dose does provide a more potent and durable response. And so they do have a backup study going on. That's the Ensemble 2 that is looking at 2 doses in case all of the patients may need that or some of the patients may need that. So we'll find out if it's going to be a 1 dose or a 2 dose vaccination for the patients once that clinical outcomes are analyzed and available for us.
Right now, the phase 3 study, whose data are due to be released any day now, that's a 1-dose regimen, right?
Absolutely. That one was completed, and that's the one that they're analyzing, and hopefully we'll have at least some of the groups potentially that will be sufficient for clinical outcomes, but they do have a 2-dose that's currently recruiting as well.
Great, great. Thank you. So let me just take 1 second to scan the questions, see if there's any additional questions before we move to the final panelist. Okay, I'm not seeing any new questions, but that doesn't mean that we're not going to be around after the presentation to answer any questions you have. Thank you, Jen. Appreciate it. Great job. Appreciate
Thanks.
it. Last but not least, I want to go to Kevin Diekhaus, who is the chief of our infectious disease division, who actually is boots on the ground, deep in the trenches, actually providing care to the patients with COVID-19. And we want to thank you as a frontline healthcare worker. You are a hero now. Well, among all of us, the researchers as well. You are advancing the treatments. And so Kevin, my first question for you is, we talk about the treatments. Very nice presentation by Adrian. Are there any medical and non-medical treatments for hospitalized patients and the outpatient setting? So Dr. Vela mentioned the cocktail of antibodies that's infused. I know you personally supervise this treatment at John Dempsey Hospital. So maybe you could tell us more about that, as well as anything else you want to talk about on the treatment.
Sure. I think, so as a clinician, I think there's first of all, there's an awful lot of information coming out about these, all of these medications, including some future medicines that we haven't used yet. And it's kind of our job to kind of synthesize that and say, okay, we have someone in front of us. How do we treat that person? And so although a lot of the studies are having difficulty showing a signal for effect, you may be able to look at subsets of patients and say maybe there's a little bit more effect in that subset of patients. And so we look at, as someone we're admitting, we look at how long they've had the illness. Early in disease tends to be more of a viral type issue, whereas later in the disease tends to be more of an inflammatory immune sort of process. And so we can target the therapies a little more directed towards that. And then there's a bunch of non-medical therapy that we can do as well. So for example, just simply proning a patient, which can be quite difficult if someone is on a ventilator, to turn them on their stomach. And that helps equalize their ventilation and perfusion in their lungs and allows for better oxygenation. So simple mechanical measures can have a big impact. In terms of the medications, remdesivir, I think, has had kind of a bad rap. I mean, it's has shown to be effective in some very limited settings. We limit that to fairly early in the disease course, while people are thought to have the more viral type of illness. But if someone comes in after a week or two of illness, it's probably not going to have that much effect. And so at that point, the risk outweighs the benefits. Convalescent plasma, which is, again, has a long history. Actually, the first use of convalescent plasma was in 1890, believe it or not, for diphtheria. So this is pre-antibiotic. Using antibodies from convalesced patients also is an antiviral type of thing. So if we get any signal from our studies, it tends to be earlier in the disease course. And so someone coming in the first week of illness, we might be more prone to give convalescent plasma. One project that was not really talked about a lot, which I think has really a good signal for efficacy, is dexamethasone. So that is a drug, it's an anti-inflammatory drug that is helpful, not so much for the virus, but for the intense immune response that the virus elicits. And so there was a large trial called the Recovery Trial with over 6,000 patients, 2,000 that got dexamethasone, recently published in the New England Journal, that showed efficacy for that. So most of our patients were hospitalized, and if they're to the point of requiring oxygenation, will end up on this anti-inflammatory. There's with the interleukin-6 inhibitors, tocilizumab being the primary example. Again, mixed signals. I think right now that's fallen off of our treatment protocols. But one thing that has gone on our treatment protocols has been a side effect, if you will, of the infection with the inflammation has been much of the morbidity has been related to the clotting cascade. And so it's quite likely that with severe disease, with the intense inflammatory response, that you can get deep venous thrombosis or pulmonary emboli, clots that move about. And that can cause severe illness. And so simply placing someone on anticoagulation can prevent that complication. A lot of other therapies are being looked at. I think there's not enough of a signal to really put them into treatment. One that you may hear about in the future, it's gotten kind of a hot topic, is ivermectin, which is a anti-parasitic agent. I won't go into great detail. There's a lot of data for and against, but it may be something that we might find useful in our extremely critically ill patients. Again, these are very early studies, and I think more information is going to have to come out about that. The monoclonal antibodies that you mentioned, I think, are very important. So those have been shown to be effective. This is basically using two different products. One is called bamlanivimab, and the other is casirivimab and indevimab. And the hardest thing about using them is simply pronouncing them. So these are basically an hour-long infusion, but it's targeted against people who are in the viral phase of their disease, so fairly early on. And it's more of a preventative. So people who are not quite, maybe mildly ill, but not severely ill, who have risk factors for developing severe illness. So things like simply age, so age over 65, an elevated BMI or obesity, chronic kidney disease, diabetes, or immunosuppressive therapy. Those are the types of patients that we want to be able to bring in and give a directed antibody to. The studies have shown that the result is that there's a less likelihood of them needing hospitalization or returning to the emergency room. So again, it's approved under an emergency use authorization, but there's fairly good data for significant clinical outcomes. But again, it's more of a preventative. It's not, once someone is ill enough to need hospitalization and oxygenation, it's not really been shown to be effective.
Yeah, Kevin, I'd just been scanning some of the questions. So one of the very good question is the vaccine can generate the antibodies. And so they are monoclonal in the sense that they are one of a kind that is directed against, say, the spike protein. And then you have artificially synthesized monoclonal antibodies, which is the one you just talked about. And so there are two different production. One is the body's own production, which is elicited by the natural infection or by the vaccine. The other one is this artificially synthesized monoclonal antibody, which you can actually make in large quantity and infuse to patients with COVID. So, so one of the original purposes of the artificially synthesized monoclonal antibody is as a bridging therapy, right? So before you can get the vaccine, you got sick. And so we're going to give you that antibody, that monoclonal, as though you already been vaccinated before. So it's a way to provide a boost from the outside. And so I saw this question, I couldn't resist, but trying to make a distinction between the two. But so are you, how many patients have you actually infused this artificially made monoclonal antibodies at UConn Health?
Right, so we've been running this program since about mid-December, and we've probably had, I'm guessing about 30, 35 patients so far. We certainly have capacity for more, and one of the, I'll put a call out, we're looking for patients. This is a very effective therapy, and if someone, certainly age over 75 is a high-risk category. We're very interested in trying to get those people infused. I did see a question come up about working directly with nursing homes, and I think that is something that will probably be happening in the future. Right now, the processes that are required for an infusion are best handled inside a hospital in case of anaphylaxis and the like, but I do think that eventually it's going to be moving to the community like that.
Okay, very good. So there was another question which is related to also my question, which is we know the vaccines can prevent disease, the symptoms, symptomatic disease. What about blocking its transmission? So if I got the vaccine, in the Pfizer and Moderna, I'm 95% protected against severe symptomatic COVID, but does it mean that I won't transmit the virus if I got the virus?
Right, so that's a really important question that I don't think has been fully answered yet. Certainly from an epidemiologic sense, if you can prevent infection with the virus, which is what the vaccine does, it's 95% effective in preventing clinical disease or laboratory documented disease, and 100% effective, at least for Moderna, for severe disease. That is going, just in itself, is going to reduce transmission. Now, whether it's possible that someone could have a subclinical or asymptomatic infection and be able to transmit, it's possible. That has not been studied, but I think on average, being vaccinated on a community basis is going to reduce transmission.
Okay, so related to that, so that means even if you are vaccinated, you should still wear a mask, right? Absolutely.
So right now, there's just an incredibly high degree of community COVID right now. It's out there. And even at 95% effective, there's still that 1 in 20. So there've been a couple of very publicized, politicians that said, I got the vaccine and I got COVID, but they had mild disease. So it's still out there. It's really important that as we get the community level down, we have a saying in infectious disease that, nobody is safe till everybody is safe. And so we want to really drive the community rates down. And vaccination is one really good way to do that. But the other is continuing, doubling down on the social distancing, hand hygiene, wearing a mask, avoiding all the stuff that we've been doing, avoiding large gatherings and that sort of thing. So for the short term, we still have to double down on all of that.
So that 1 out of 20 that did not seem to be protected, what kind of symptoms? Did they also have severe symptoms?
So the breakthrough cases, first of all, many of the breakthrough cases, and we're seeing this a little bit, people are getting a single dose of vaccine, but the studies really define this as 2 weeks after the second dose. We're in the midst of a vaccine push right now, and we are seeing people that are getting their first dose of vaccine and probably incubating coronavirus and coming down ill. It's an issue. It's possible that it's a 95% likelihood after 2 weeks of disease, but in terms of severe disease, it's been 100% in the initial Moderna trial. So it's mild or asymptomatic.
Right. Okay. Got it. That's very helpful. So one other question for you, that is, we heard about the UK variant, which has got 6 or 7 simultaneous mutation, the virus engineered themselves. They try to survive and they try to proliferate. And then the South Africa variant, which has additional mutation, I think 3 additional new mutations on top of the mutations that the UK variant has, which is causing more concern, I think, at this point than the UK variant. So can you tell us more about what can we do about that, and what about the vaccine that we have right now and how effective they are against these 2 variants?
Right. So I'll actually make it a more common. There's a 3rd variant that we're worried about, which is coming from South America.
The Brazil one.
No, the Brazil variant. So this is what viruses do. They evolve over time. In our field of HIV medicine, this is a major clinical issue for us in HIV medicine, but these evolve. The concern is that the evolution with these changes is they can increase the transmissibility. They become a little more fit. They can possibly increase the morbidity or the disease, or the others with the changing of the epitopes, that spike protein. It's possible that vaccines could be less effective. So those are the concerns. And I think with each of these viral variants, we need to evaluate each of these concerns. So the UK variant, or the B117 variant, it really became prevalent in Southern England. And we've had, I think, 8 cases in Connecticut now. It's expected that this variant is actually going to become the dominant variant in the United States by springtime. And it is more likely to be transmitted in certain circumstances. So it's 50% more likely to be transmitted than the virus that we're dealing with right now. So there's a real concern for that. There's also a little bit of a signal perhaps for increased disease severity. But fortunately, looking at serum from people who have been vaccinated, it seems to work against this particular viral variant. So vaccine seems to be okay for this variant. Not so much for the South African variant. So this is a spike protein variant as well. It's, that the Moderna company looked at people who had been vaccinated with their particular vaccine and noticed that there was a significant reduction in the neutralizing ability of their serum against this particular variant. So that's concerning. Now, that's being said, it was reduced, but it was still at the level that it was considered effective or efficacious. But the advantage of these new technologies of vaccine is you might be able to tweak that vaccine a little bit as the new variants come out. So this is very much an evolving story, pardon the pun, but this is a very changing story. The Brazil strain, which I think is kind of the newest, it just recently was recognized in Minnesota. This is the strain that really has devastated the Manaus area in central Brazil. It's definitely more transmissible as well. And we really don't know that much about it in terms of its disease or the impact of vaccines on it. So, we're going to be finding out more as time goes by.
Thank you, Kevin. So we do have quite a few questions here. If the panelists are okay, if you could stay on for a little while longer and you can unmute so that this can be directed at any of the panelists. So the first question is, does an allergic reaction to contrast dye prohibit receiving the vaccine? We all heard about the allergic reaction after vaccine. Perhaps Jen and Kevin, you could share your thoughts on this question.
Absolutely. So that's not one of the things that is a usual contraindication for someone to get either the Pfizer or the Moderna vaccine. The Center for Disease Control and Prevention has been doing an amazing job of keeping up to date about all of the specific contraindications and how to manage patients with different types of allergies. So if you have specific other questions on that, it is on the CDC website, and you would be able to look there and determine if you could get the vaccine. Obviously, if it's not obvious, talk to your healthcare provider, and they'll be able to help advise you, or tell them when you go into the vaccine appointment, they will ask those questions, as they do need to follow those specific parameters on who can get the vaccine, who cannot, and then also how long you have to be observed for, whether it's the 15 minutes that's standard, or there are some patients that can get vaccines that would just need a little bit longer term.
Thank you, Jen. Kevin, you have anything to add?
I think Jen did a good job with that. I think, we're learning more about these vaccines all the time. These are safe vaccines. The adverse events that were highly publicized, you have to remember, we've given out 22 million doses of vaccine. So you're going to have some adverse events that are going to be reported. And I think the media is very sensitized to be reporting that. So again, what is a signal and what is, kind of background is hard to say. But the rate of severe reactions has actually been quite minimal.
Yeah, something I read is 10 in 1 million. 10 allergic reactions in 1 million. So very low indeed. This is a question, maybe either the Adrian or the Kevin, if someone who is immunocompromised, such as with a chronic lymphocytic leukemia, does that qualify to get the monoclonal cocktail?
One word answer,
Maybe
yes.
Kevin? Yeah.
So assuming if you come down with COVID, yes, give me a call.
Okay. What about factors predisposing to reinfection? That means if you got COVID, I think you can get it again. Not very frequently reported, but do we know anything about predisposing to reinfection? Maybe back to you again as the infectious disease specialist, Kevin.
Right. So surprisingly little. There's basically a series of case reports, and in fact, we had a case here that I'm convinced was reinfection as well. There hasn't seemed to be any particular consistency between them, and I think again, more to come. There's so much that we don't know about this virus in terms of what predisposes. I suspect that the answer is going to be hiding in immunology, but again, that data is not really out there.
Yeah, maybe I can ask Tony if he's aware of any published or unpublished report on immunology, immunity, and reinfection.
It's a tricky question because it really would depend on the state of an individual's immune system. So I think, most of us immunologists pride ourselves on the fact that you have memory. And so that if you do get reinfected, you're able to handle the infection and perhaps not even know about it. But there could be examples where individuals would have a compromised immune system and then maybe be able to get the same infection, with the virus. So I think there's much more to do. And one thing that hasn't been mentioned in this panel is that we know very little about this. I mean, we're talking a lot, and we do have a lot of studies, but in reality, we know very little about what's going on and how this is going to turn out in the end. And it's very important to keep up all the basic research so that we could answer more specific questions such as reinfection and why did some people get reinfected? Did they have perhaps a defect in their T cell response that allowed them to get reinfected while maybe another individual did not? So we have a long way to go in understanding many of these intricate questions. And the only way we're going to do it is by pushing science first, especially basic science that will lead to translational and clinical studies, and then drive the treatments from there.
Thank you, Tony. I'm going to group the next three questions on vaccine together. Why are so many people skeptical about getting the vaccine? Is there really that there will be a long-term side effect from these vaccines? So that's one. If you are taking your medications, do you stop the medication after you get the vaccine? And the third one is, what if I have autoimmune disease, right? So my immune system is already kind of jazzed up. Is it safe for me to get the vaccine? So I'm open this up to anybody who wants the answer.
Bruce, I'll talk a little bit about the fear of getting vaccinated. One thing that's really not out there in the lay public is that vaccines probably are the single most productive medical treatment that we've ever had in the history of humankind. It has saved millions upon millions of lives. And I guess memory, some of our memories might be short, but you go back to polio, go back to many examples of how vaccines have saved literally millions of people's lives. With everything, there's going to be side effects. I mean, there's not a single thing out there that I know of where you can't have an allergic reaction to one thing or another. But a lot of that history of skepticism with vaccines was built on false data suggesting that they caused autism or other types of diseases that were proven to be untrue. And I think it's really important, again, to have science drive fact from fiction. I think Jen could probably have more to add on this story as well. So I'll stop there.
Absolutely. I agree 100% with what Dr. Vella mentioned. I think the other thing that's specific to these vaccines is that people don't necessarily realize how many thousands of patients were included. They think that this was ultra fast, but I think it was ultra fast because it is technology that we can use. We had the sequencing of the virus early. We also had a unique situation where the government and pharmaceutical industry and almost unlimited money all combined together to really study this focus. So if you look at the number of patients who were included in vaccine trials from 2000 to 2010. This is around the same number of patients that were included in all of those, from Prevnar to Prevnar 13. The only vaccine that had a lot higher was the rotavirus vaccine, and that was a unique situation. But for these vaccines, they are similar to Boostrix and your Tdap vaccines, Prevnar. It's got the same number of patients. So I think people need to know that, yes, it's only short-term data that we have at this point, but we actually did study this in tens of thousands of patients. And now, as Dr. Diaz had mentioned earlier, over 20 million doses have been given in the US. So we have millions of additional patients, and most of them healthcare providers, and we provided information to the CDC in a new real-time vSafe documenting ability. So we're getting real-time data that the CDC has analyzed, and that's how we know that it's 11 cases per million that are having anaphylaxis. That's how we have those data, is because of how quickly and how real-time these reporting are going. I was a little bit, until we started to see that phase 3 data and those great studies in the New England Journal from both of the Pfizer and the Moderna and how efficacious they were, many of us didn't know what the data looked like, and now we do. And I think that those are some real important points, that we have the same numbers of patients that we always have, that they have such great outcomes. And as Dr. Diaz mentioned earlier, 100% efficacy for the Moderna against severe clinical disease. Taking that versus the thousands of patients dying every day. This is so super important to get our patients vaccinated, and we really need to explain that to them. So I think that's one thing that really trying to get that message out to our patients is super important.
And I'll add,
Thank you, Jan.
there's a
Great point.
there's a number of kind of
Go ahead, Kevin.
narratives that are out there that I think we can work against. So there's a narrative that it changes your genetic code. It doesn't. It's an mRNA vaccine, but it's not integrated into our genetic code. So we're not going to change your genetic code by giving you a vaccine. There's a narrative that it causes infertility. It's not been shown to do that. There's even a narrative that Bill Gates has a microchip that he's going to chip you when you get vaccinated. And that clearly is not true as well. So there's all sorts of conspiracy theories and narratives out there. And I think as scientists and as educated people, we need to educate the public that this is scientifically sound and was developed in a very expeditious way, but also in a scientifically rigorous way that maximizes patient safety.
Okay, maybe to you, Kevin and Jen, whichever one, if you have autoimmune disease, can you still get the vaccine?
Well,
That's
a
not
good
an
question.
exclusion, right?
It's currently not an
Huh?
exclusion. So I'll take that. It currently is not an exclusion. There have been some discussions in a theoretical way of a concern. I think the data, this is something that the data is going to come out and describe. At the moment, it is not a contraindication, and certainly the risk of with the community activity of COVID right now, the risk of COVID probably outweighs the risk of adverse events related to the vaccine. I'm curious, Jen, if you have any comments on that too. This is one of those areas where there's more questions than answers, and I think the data will eventually fill in the gaps.
You know what? I agree exactly with what Dr. Dekas has said, that the CDC has clearly said we don't have the evidence. It wasn't a condition that was excluded from clinical trials. We didn't see any specific signals, but there's always the potential. And so we just don't have the evidence on that population yet. So it's a risk versus benefit situation. Most likely, it's probably fine, but.
I think I can handle the next maybe two questions just in the interest of time. So monoclonal antibody cocktail that's infused for patients with COVID. It's not offered everywhere, but I would say, major hospital systems such as UConn Health, in this case under the Kevin Dekas, has been infusing it for over a month now. And so that's available, and there's actually plenty of supply. So if you are qualified and eligible, please contact Kevin. I'm not trying to be his PR manager here. The next question is that I had a cold. I thought that maybe I had COVID, but I got tested for the antibodies against COVID. So this is something I know about because we're studying the antibodies against COVID-19. No, if you don't have the antibody, then you were not exposed or contracted the virus. And so this is a question for Jan and Kevin. I received my first shot and I'm supposed to get the second shot next week. What will happen if there's a shortage? I can't get my second shot after 21 days. Does that ever happen?
So luckily in the state,
here, right?
not here.
Not here.
Luckily in the state of Connecticut, there's first doses and there's second doses. So if you got your first dose, we're fine at this point in time. Like everyone else is saying, everything is constantly evolving. So I can't guarantee that to be forever, but for right now, if you got your first dose, there is a second dose that will be available for you.
Yeah, related to that, if I got my first shot on Pfizer, if there's a shortage, should I get the second shot with Pfizer? But if there's a shortage on Pfizer, can I get it from Moderna?
So I'll take that. The easy answer is no. These are meant to boost each other. And so you do need to use the same product for the first and second vaccine.
Okay, so this is another question. I think maybe Adrian and others can weigh in. How does it take for us to control a pandemic with both vaccines and maybe treatments like drugs? How long does it take? Any projections, speculation?
No, I don't have an answer for that. Treatments so far are most of existing treatments are not useful for COVID-19. The most promising, as Kevin mentioned, are monoclonal antibodies, and still are some ongoing trials for early treatment and prophylaxis, and they are probably coming in the next few months. And the answer now is vaccines. So is the most important intervention in the next few months, but a given time, I don't know.
Thank you, Adrian. Jen or Kevin, do you have any projection as to when the vaccine will achieve herd immunity? I think maybe that's the underlying theme of the question.
Right.
Herd immunity.
So the estimates are that 80 to 90% of the population needs to be immune, and that's immune either by vaccination or by natural disease. Unfortunately, we've made strides towards that with natural disease, unfortunately. With the vaccine efforts that are undergoing now, the estimate is, we'll probably be somewhat out of the woods, so to speak, by the end of this current year. But again, anytime we've predicted the future, we've been proven wrong. So I think we just need to watch and wait. That also doesn't, that's for the United States. I mean, this is a worldwide illness. And I think, I'd just like to raise the issue that, allowing vaccine use in the developing world is going to be just as important in protecting that population, just even for our own sake, in terms of reintroduction of disease back into the United States. So this is a worldwide issue. It's not just parochial to the United States.
I think the other thing that's really important on that is that about a quarter of our population, this is my other passion, so a quarter of our population is pediatrics. So it also depends when we can get the vaccine for our pediatric patients approved as well.
Okay, so what about the blood type? I have type A. Does it make me more prone to have the infection?
There was a large epidemiologic study that looked at type O versus type A, and there was a tiny, tiny signal that those who had type O fared a little bit better, but it's certainly nothing that I would hang my hat on. So if you look at a large epidemiologic study, there has been shown to be a statistically significant difference. Clinically, though, that difference is very, very tiny. So I wouldn't bank on your blood type either protecting you or dooming you.
Okay, I'm going to answer the next one, which is the transmission by touching a surface. So a simple breakdown is, I think, almost half is by airborne and half by aerosol. And maybe only 5–10% by touching surface. And that's kind of the general thought. So most of the transmission occurs by just talking or breathing without the mask. All right, so here's another question. So if there's a shortage, right now it's 3 or 4 weeks between the first and second shot. How long can we stretch out that window? Can we go to 6 weeks between the first and second shot and still make the T cell response after the second shot, make the antibody level goes up after the second shot?
Sure. So obviously, the earlier we can get our patients vaccinated on schedule is best, because that's what's been studied. The CDC has come out and said that you could schedule, if you have unique situations, up to 6 weeks out, but they don't want to do what is happening in the UK right now and going 3–4 months out, because the studies haven't been done. We don't know for certain, and that's a significant delay. We don't want patients to have significant lack of efficacy due to a single dose and come down with significant COVID disease.
Okay. Anybody who wants to weigh in? I think, Jen, you did such a good job. I think we're satisfied. Here's another question. JAMA had an article just 2 weeks ago saying that 50% of the new infection came from people who were asymptomatic. Do you guys agree with this conclusion? Have you researched on this topic yourself? 2 questions. Do you agree with this conclusion? Half of them came from asymptomatic spread.
I'll tell you, certainly a large number of them do. So whether it's 50%, 50.0% is hard to say, but and this is an example of how data and information has changed over the course of the pandemic. Early on, if you recall, it was okay not to wear a mask and it was surfaces only. But as data has become available, we realize that there is asymptomatic disease. That asymptomatic disease is likely, you're very likely to transmit, and it goes to 48 hours before onset of clinical illness. And so contact tracing is going to be key in terms of, identifying who was around you and within, 6 feet or within an enclosed space within that 48 hours prior to onset of illness. So yes, a majority of disease or a significant part of disease is transmitted from asymptomatic people. And that is why it's really critical that we maintain social distancing and wear our masks, because you don't know if you're the person who's asymptomatic and spreading it, or if someone else is asymptomatic and spreading it to you. So while the community rates are so very high right now, it is critical to keep the social distancing pressure up.
Okay, so the next question, maybe Jen or Kevin also can answer. What about timing? My parents could not get an appointment. I think this refers to vaccine in the 3-week time frame. So maybe this speaks to the sort of the shortage we're facing, or just the rolling out process.
So I'm not sure I quite understand the question. The systems are set up that at the time of first vaccination, you should, at least in Connecticut, have that second vaccine carved out for you and a process in place to make that second visit. So I'm not sure about the time frame. These are timed very carefully, and I know at least at our site, if you get your first dose, at the time of your first dose, you have an appointment for your second dose and you have guaranteed vaccine for that second dose.
Okay, great. So if you have any question, email Kevin Decaus. just tongue in cheek. So the next question is, how long do the current vaccines, if someone already has severe COVID and recovered, do they still need the vaccine? And if so, when?
So this is a little evolving as well. It's clear that we don't want to give the vaccine during acute illness or during the period of isolation. Some people would give it soon after infection. Other people would wait a little bit. But it used to say some patients may want to wait till the end of a 90-day period. And earlier this week, the CDC has changed that phrasing a little bit because of the concern of potential waning immunity as it gets closer to that 3-month period. So patients can get the vaccine as soon as they're clinically able. And if they want to wait a little while after having acute COVID, that's fine. But the longer duration that they wait, out to 2 months or 3 months, they could potentially become susceptible again.
So I just saw a question that disappeared. I think the question says, how long is the vaccine protecting you for? We may have mentioned this in the initial dialogues, but what is the data out there?
I mean,
3 months, 6 months?
I can answer that real quick. I mean,
Tony, yeah, go ahead.
Yeah, we just don't know the answer to that for the COVID vaccines. So, but I would encourage anybody that's interested in that type of question to really look at the New England Journal Medical the paper from the Pfizer vaccine, because a lot of the details and the time points, and they're going to be following up on that study as well. So it's just premature to answer that question right now. But if you look at the graphs, and you can see that it's a very robust, looks very durable, and I think, you know, probably in the next few months we'll get more and more on how the vaccine, at least the Pfizer and the Moderna vaccine, pans out time-wise.
Thank you, Tony. I think I would concur with that. I think time will tell. And that's why you still have to wear the mask and practice precautions, social distancing, washing your hands. Down to the last 2 or 3 questions. Or there's more actually just popped up. What condition have to be met? We're happy to answer them. What condition have to be met for us to say that there is no longer a pandemic in Connecticut? I mean, maybe the infection cases, maybe the positivity rate. Which metric would you use to say we got this under control? Kevin, maybe?
I may not know the exact definition of the pandemic. Pandemic means that there's spread basically more than an epidemic kind of across the world. So our local epidemic, one of the things that we look at closely is called the rate of transmission, or the R naught. And of course, with all the social distancing, we want to get the rate of transmission down significantly. And I think that's a primary driver of this. But of course, with the decreased R naught, you're going to have decreased numbers of patients. Basically, it's a mathematical way of defining transmission rates. And when we have control, that's when the pandemic will be declared over. Unfortunately, I do think this is a virus, you know, Pandora came out of the box. I think we're going to be dealing with some aspect of this virus for a very long time to come.
Anybody else want to chime in? Okay. If you have a choice, which vaccine would you pick? Jan?
Whichever one you can get. Right, they both have about 95% efficacy. You're not going to have a choice. You know, if you're if you're one of the highest risk groups, your name comes up on the list, go and get your vaccine.
I would agree. Okay, the next question is how about double masking as a recommendation? Double masking, wear 2 masks rather than 1. Would you recommend that?
I think, so that question just gets to the quality of the mask, really. I mean, so there are recommendations in terms of using a cloth mask. And the cloth mask, the pore size should have a double layer to it, just to reduce the likelihood of aerosolization of the virus. The standard medical mask, surgical mask, should be adequate by itself. And again, for certain situations, you know, in a medical sense, we have an N95 mask, which is much higher. And so I think it depends a little bit on the exact circumstance, but I wouldn't necessarily wear a double medical mask. The masking, so for example, the bandana type that is a single layer of cloth is definitely inferior to a standard cloth mask that has more than 1 layer. It is recommended to have more than 1 layer on a cloth.
Okay, we only have time for one more question. I think back to Jen. Is the J&J vaccine a live vaccine?
So no, it's not a live vaccine. It's a non-replicatable adenovirus vaccine.
Got it. Thank you. There's an HIV question, maybe very quickly for Kevin, since that's one of your areas of expertise. Now, I don't know if this is the right, but it takes longer for the second dose to be fully effective for HIV clients. So if you are HIV positive, does it take longer for the second dose to take effect?
Right, so it's a really good question and one that we have no idea what the answer is. So I think as data comes out on vaccine and HIV positive individuals, and this is coming up in my clinic all the time, the concern with people who have underlying immunodeficiencies, be it HIV or a medical condition or some of the treatments that reduce immune response, is just simply not having as much of an immune response to the vaccine. And again, I keep harping on the community, but this is where we need to do community vaccine, because it's not only the person you're vaccinating, but you're actually protecting the people around you. So we need to have the community rate go down to protect people who don't necessarily respond quite so well to the vaccine themselves.
Well, thank you. That rounds up the entire panel. I'd like to thank each of the panelists for such lively discussion, such great information. I'm seeing on the chats a lot of appreciation, and thank you for your informative answers. And also, I want to thank Liz Kruger from the UConn alum and for organizing such a wonderful event. Even at 6:30, we have 65 people still attending. So hopefully you will join us for the next panel in February, and we were going to close out this series in March. So once again, thank you very much, and you all have a safe period ahead and a wonderful evening. Thank you.
Thank you.
Thank you.
Bye-bye.